Welcome back, everyone. So we're talking about gene regulation in eukaryotic cells. We've talked about things like how cloning is done, how we know that all of our genes are present in all of our cells. We've talked about how our B cells are able to change their different genes around for their antibodies to produce more antibodies than what you would expect just by the number of genes we have. Next, we're going to talk about some different ways that we can control transcription. So taking our actual genome and turning some genes on or off. One of those is by DNA methylation. So here's an example of a cysteine on your DNA. You can see there's no methane here, but here there is a methyl group. That methyl group, that is what methylation is. Just adding that little tiny little tag onto that cysteine is going to tag that gene, generally to tag that to turn it off. So we can methylate or add those methyl groups to cytosines in the DNA to reduce that gene activity. So in cells where that gene is going to be down expressed, you would expect to see more methylation of that gene. Especially mammals, eukaryotes, but especially mammals, rely on DNA methylation just in a broad sense to regulate how accessible a piece of DNA is to the transcriptional machinery. So if this is your DNA, and in the promoter, there's going to be probably CPG islands or lots of cysteines and guanines. And if those are methylated, which is what these little red tags indicate, these transcription factors aren't able to bind, which means that that gene isn't going to be expressed. You are more likely to have these proteins that bind methylated DNA bind. So, for example, is MECP2. And that is also going to affect then how histones and chromatin are being remodeled to not only have that gene be methylated, but also you'll end up with impacts in how that DNA is being wrapped around histones and how that DNA is being wrapped into chromatin. So overall, you end up with this net transcriptional repression. So these DNA methylations tend to be clustered on the 5' ends of genes. Methylated cytosines are typically next to a guanine, so that's where that CPG came from. You have a cysteine, a cytosine, then a phosphate, then a guanine. So you have these CPG islands, which are there to be able to be methylated to be a region of control for that gene. You can end up with either a localized or a regional silencing of gene expression, silencing either just one gene or silencing a large piece of that chromosome. And those DNA methylations block the binding of transcription factors, can change how chromatin is being packaged, and that also will deny access of the transcription factors to the promoters. Another way that chromosomes can be inactivated is found in cells that have more than one copy of an X chromosome. So the typical genetic profile of a female mammal is to have 2 X chromosomes. Typical pattern in a male mammal is to have an X and a Y chromosome. Although there are some differences there, that is the typical pattern that you expect to see. So men are able to have healthy functional cells, which tells you you only need 1 copy of your X chromosome for your cells to function. And actually in women, in our cells, we don't have 2 X chromosomes being expressed. One of those X chromosomes is going to be inactivated. So during embryonic development, early during embryonic development, one of your X chromosomes is going to be randomly inactivated. It may be, so here's some different pictures showing, you have a chromosome from mom and from dad. It may be dad's chromosome in some cells is inactivated. It may be mom's chromosomes in some cells is inactivated. It's going to be random which one is picked out. And in that X inactivation, that inactivation of that X chromosome, the DNA on one of those X chromosomes is going to have a lot of methylations on it. And that extensive methylation is going to trigger the chromatin to condense. And you end up with this tight mass. It's called heterochromatin because it's so tightly wound up. And you can see in the picture, it instead of being a nice, long, accessible X chromosome, you have this tightly bundled up little X chromosome. That bundled up inactivated chromosome is visible. If you were to look at that cell under a microscope, you'll see a little dark spot. That's called a Barr body. So if you see a Barr body in a cell, you know that there are 2 copies. That is an inactivated copy of the X chromosome. Then once that X chromosome is inactivated, anytime this cell reproduces, that same X chromosome is going to be inactivated. So all of the daughter cells produced from that early embryonic cell will all have that same pattern of X inactivation. So overall, you'll end up with cells in an adult female and in an adult male, or in a female or in a male, that will have 1 active X chromosome. Regardless of how many X chromosomes are there, only 1 is going to be active. So that means in a female mammal, in a female placental mammal, our cells will be a mosaic. We will have some X chromosomes activated in one cell, while other cells will be inactivated, and other cells will have the other X chromosome inactivated. And you can see this random pattern show up in calico cats, or tortoiseshell cats. That's what this cat is. Their fur color is an X-linked gene. It's on their X chromosome. And so in this cat, some of her X chromosomes have ended up with the orange gene being inactivated, which means the black fur gene is going to be expressed. In other of her cells, the black fur gene has been inactivated, so the X chromosome with the orange fur is going to let that orange fur color be expressed. So she has patterns where her fur is black and patterns where her fur is orange. So typically calico cats are going to be females. They're going to be cats that have more than one X chromosome to have that tortoiseshell patterning to them all because of their random inactivation of their X chromosomes. The ways that X chromosomes are inactivated is using a pattern of RNA called Xist RNA. Xist RNA is a long non-coding RNA, so it'll be transcribed from your X chromosome, and the chromosome that it's transcribed from is a chromosome that's going to become inactive. So once a chromosome starts transcribing that Xist RNA, it's going to become largely inactive. And because it is non-coding, that RNA is not going to become translated into any protein. Its function is to engage the inactivation of that X chromosome. So that Xist RNA then is going to start coding that inactive chromosome. So you can see this chromosome has been selected for inactivation. It starts making its Xist RNA, and then it's going to start coding that X chromosome with it. And once that chromosome has started getting coded with that Xist RNA, it brings in these proteins. These are chromatin modifying proteins. So they're going to take that chromosome and condense it into that tiny little Barr body. So you'll see in this picture, you can see all these chromosomes in their cell, and then this Xist RNA coding this chromosome. It is getting ready to be condensed into a Barr body. You've probably heard about epigenetic changes. That's something that's been in the news a fair amount in the past few years. An epigenetic change is a stable change in gene expression. By saying it's stable, that means that if you have a gene that's been epigenetically modified, it can actually be passed down to your children and to their children. It can stay that change, causing that change in gene expression for a couple of generations. But in an epigenetic change, you have not actually changed the DNA sequence. What you've done is you've done something like methylate your DNA, and it's done so stably that it can actually be inherited. You hear a lot about epigenetic changes as something that may affect someone who lived through a childhood trauma, so someone who was a child during the Holocaust, and that trauma caused this epigenetic change in their DNA that can be passed on to their children, and their grandchildren will still show impacts of that trauma in their patterns of DNA expression, even though they didn't survive that trauma themselves. So those DNA methylations are inherited and will impact the gene expression of your children and grandchildren. These epigenetic changes are also responsible for something called genomic imprinting. Genomic imprinting, that means that if you inherit a diseased gene, what that disease looks like can be different depending on if you got that diseased gene from your father or from your mother. Not all diseases have that pattern of imprinting. One that does is Prader-Willi or Engelmann syndrome. So on your chromosome 5 or chromosome 15, if there is a deletion of paternal genes and the maternal copies are methylated, you end up with Prader-Willi syndrome. If you have the opposite, the maternal genes are deleted and the paternal gene is inactive, you end up with Engelmann syndrome. And these are going to be different, where Prader-Willi is a fairly intense disease. Engelmann syndrome is even more intense, if I can say that. A little more intensive of a disease. And you have these two very different patterns of disease just because it's either the maternal copy or the paternal copy that is causing that disease. Another type of epigenetic inheritance is histone modifications. So this is when we take histones, which are the little proteins that we wind the DNA around, so you don't have DNA just hanging around in your cell getting tangled up. Our DNA is wrapped around these histones, and you can change those histones by either methylating them, putting a methyl group on them, or acetylating them, putting an acetyl group on them. And these modifications to your histones are also heritable. And those post-translational modifications that you make to histones can be rewritten onto newly assembled nucleosomes as the cells are copied. Okay, so we just discussed a few different ways that cells can basically turn genes on and off to impact what genes are going to be created. We still have a few more things downstream, talk about how we control transcription and translation, and I'll see you for that. See you later. Bye.